Pyrolysine was the 22nd amino acid to ever be seen, and its discovery paved the way for the incorporation of unnatural or non-canonical amino acids into protein. So, pyrolysine is this weird amino acid or protein letter that's used by some bacteria and archaea. So, these kind of like bacteria like things that often live in extreme environments. And in the early 2000s, these scientists found that there was this archaea that was making this, that seemed to be skipping over a stop codon, so skipping over the instructions that the bacteria, that the ribosome, the protein making machinery, uses in order to tell it which amino acids to add in what order. So normally there's a stop codon. When there's a stop codon, when the stop codon appears, the ribosome knows, okay, this is the end of the protein, let's cut this chain off. But what was happening in these bacteria or these archaea was that they had a stop codon in the middle of like their full protein. And when they investigated it, they found that instead of stopping, the ribosomes were actually incorporating a weird amino acid. And it wasn't one of the like normal amino acids, like the 20 canonical amino acids we think about. It was this weird one. And they had to do a little work to figure out what it was, and we'll talk about this. But they discovered it was this molecule called pyrolysine. And what's more, they discovered like the genes that could be used to make pyrolysine and incorporate this pyrolysine into the proteins. And it turns out that this pyrolysine incorporator, basically, the tRNA is going to, the ribosome is going along. The tRNA is going to bring the ribosome which amino acids to add based on the sequence of the messenger RNA that the ribosome is traveling along. So the ribosome is traveling along the instructions for making the protein. The tRNA is bringing it the corresponding amino acids. And so this pyrolysine has its own tRNA. And its own tRNA synthetase, which is going to load that tRNA with pyrolysine. It also has the genes for making pyrolysine, but you can actually just put in the loader and the tRNA, and now what happens is that you can get, if you add in pyrolysine already made, or if you add in something that maybe looks like pyrolysine, you can get the ribosome to incorporate it at a stop codon, even if those cells wouldn't normally do that. And so we call this like amber suppression in this case, because the stop codon in this case is what we call the amber codon, which is UAG. There's several different stop codons that we can talk about later, but this is the amber codon that this pyrolysine uses. And so by using this amber suppression, well, now you can get cells to incorporate pyrolysine, or you can get them to incorporate like all sorts of different things. So this paves the way for the incorporation of all kinds of different chemical groups into proteins, allowing us to do all sorts of really, really cool stuff. And so this is related to the idea of bio-orthogonal chemistry that we talked about in my post on the Nobel Prize and Carolyn Bartozzi. Basically, you're doing reactions inside of cells that aren't interfering with what's happening inside the cells. And it's kind of like happening in harmony with what's going on in the cells, but allowing you to do chemistry inside of cells. So it's really, really cool, and we'll talk more about it. But basically, you can do all sorts of different things. You can make modifications to amino acids. You can make add labels. You can do all sorts of things. And so it's really, really cool. And what's really, really cool is that you can specify exactly where in a protein you want those changes to be made, rather than trying to just like, oh, we'll stick in a modified version of lysine and now all the lysines will be modified. Here you would be able to modify just like a single spot in a single protein. And of course, there's always complications, and it's not as simple, and you have problems with like suppressing where you want there to be a stop codon. But thankfully, the UAG has a stop. It's not used that often, and so you're not going to like kill the cells too much, at least, by incorporating this amber suppression into cells that normally would think that amber codon was a stop codon. But it also means that you get low yield because, well, that stop codon is still competing with the release factor, so it normally binds there and releases the peptide. So we'll get way more into this, as well as how this pyrolysine was discovered, because it was some pretty cool science, and so I'll walk you through some of the key papers and things like that. But let's step back and talk about this from the beginning. There are 20 main amino acids, these like common amino acids that are found in all the different organisms. And So we typically think of these as having 20 amino acids. And I should say too that we're talking here about proteinogenic amino acids. So ones that actually are incorporated into proteins. So there are some amino acids that are technically amino acids because they have an amino group, this nitrogen and hydrogen group, and a carboxylic acid or a carboxylate group. And so we can call them amino acids, but we don't incorporate them into proteins. But there are these 20 main ones that we incorporate into proteins. And we say these are genetically encoded because the instructions for placing those amino acids is written in the instructions for making that protein. So when we talk about instructions for making a protein, we have the gene, which is written in DNA. That's like the original copy of the recipe. And then you get messenger RNA copies of that made, so like RNA versions. And that's a process called transcription. And then in the process of translation, that's when you actually make a protein based off of that recipe copy. And in this process of translation, the ribosome is going to travel along. And how it's going to know what to add is that you have these 3-letter RNA words called codons. And so the ribosome is going to go codon by codon by codon, and a tRNA with the matching or the complementary anticodon. So its base pairs with that codon is going to come and it's going to bring the corresponding amino acid. And it's going to keep doing this until you reach a termination, a stop codon, and then this is going to get released, except in the case where we get suppression, as we'll see. But for all those 20 amino acids, you have at least one codon is going to spell for it. And so some of these have multiple codons that will spell for it, but there's at least one. And so we say that these are genetically encoded. We can also modify amino acids after they're incorporated into proteins, and we call these post-translational modifications. But those are added after translation, so they're not like genetically encoded. They're not added that way. And so when we talk about the common genetically encoded amino acids, we're really talking about those main 20. Yesterday, and that's why for this whole thing, I have this whole 20 days of amino acids thing. But then yesterday I was like, oh, plot twist, we have a 21st on solenocysteine. And so I talked about how solenocysteine is used in these rare cases. And it does so by another kind of stop codon suppression, but it's more complicated, and I'll talk more about it so we can compare it to pyrolysine later. But now I want to talk about pyrolysine. So unlike solenocysteine, we don't have pyrolysine in our bodies, but a couple of organisms do, including some bacteria and some archaea. And so archaea are kind of like bacteria-like. They're like little single cell type things, but they have some properties that are more like eukaryotic cells. So basically, eukaryotic cells typically have cellular nuclei in these membrane bound compartments. And these archaea are kind of like evolutionary biologists have been trying to figure out exactly what the connection is between these different groups. But you can just think of them as little cell things that live in weird places. And so some of the weird places that they live involve them in places with a lot of basic things that can be used to make methane. And so there are these methanogens that are able to actually change this methane, produce methane from various methylamines and stuff. But basically they can break down waste to make methane. And so this is why this makes them useful for some things industrially. So they're studied for industrial purposes, as well as for their basic science. And it's this basic science trying to understand what's happening inside of these little buggers that actually discovered that these had this pyrrolysine. And so the pyrolysine plays key roles for them there. But really what we care more about normally is how we can actually hijack the machinery that's used to incorporate the pyrolysine. And so we're going to talk more about this pyrolysine machinery. And what's really cool about this pyrolysine machinery is that it's a lot simpler than solenocysteine. So as I mentioned, we have selenocysteine, and we have just a handful of proteins, like 20 or so proteins that actually incorporate selenocysteine. And these proteins are really, really important because, as I talked about in yesterday's post, selenium has a lot of really cool properties. It's able to form kind of like weak bonds. It's more reactive than a sulfur. So it's more reactive than selenocysteine is more reactive than cysteine, which has a sulfur. But it's able to form kind of like reversible bonds. And this is able to break up like sulfur cross links and sop up reactive oxygen species and things like that so they don't cause problems. So selenocysteine is really important, but it's not incorporated very much. And so in order to actually sneak it into a protein, you need a lot of different factors. So basically in the instructions for making that protein, downstream of where the protein making, where all those codons are, there's some like regulatory information. There's like 3' UTR. And there's this structure in the 3' UTR of selenoproteins that has this thing called a cis element, which is basically this RNA folds up into this structure, and that binds this SBB2 protein, which then binds, it brings in this a special elongation factor. So the elongation factors are what actually take the tRNAs to the ribosome. So selenocysteine has to use a special elongation factor to bring it to the ribosome, and it kind of stays right here nearby with the help of this element binding to this protein and binding to the elongation factor into this tRNA, and it kind of keeps things nearby so that you can sneak it in when the ribosome reaches that stop codon and it pauses. You can get it to sneak in the selenocysteine, so you can go past this, so you can incorporate selenocysteine at this UGA, and then when you reach a real stop codon, the release factor will bind. So that's a lot of different things that we need in order to just sneak in this one amino acid. And you also need a lot of different proteins in order to make that selenocysteine. So there's a lot of work involved in making this selenocysteine because, for one, selenium is really reactive, which makes it useful, but it also means that we have to kind of take care when our cells have to kind of chaperone it around and be really careful that it's not going around free floating and reacting with stuff. So there's a lot of different enzymes, so reaction helpers or speed uppers that are involved in making selenocysteine. And then you need all the stuff to incorporate it. What's really cool about pyrolysine is that, well, all you need is a few different things. So in these organisms, they have these 5 genes that can make it so that they can make pyrolysine and incorporate it. And if we want to incorporate something other than pyrolysine with the pyrolysine machinery, we don't even need those genes that you use to make the pyrolysine. Instead, you just need the pyrolysine tRNA and the pyrolysine tRNA synthetase. And then, as we'll see, you can kind of like evolve these and in the lab and try to find ones that can allow you to incorporate other amino acids. And so this is where we get this thing of non-canonical amino acids, and we're able to add all sorts of different things. Before I move on, I should mention that the reason why it's so much simpler to add the pyrolysine, why you don't need all of this extra stuff, in the case of the selenocysteine, we had to keep everything nearby. And so we had to use this element in the RNA, and we had to use this protein and this special elongation factor and all of this stuff, keep things nearby so that you could sneak in there. In the case of pyrolysine, the organisms that use pyrolysine naturally, they typically don't have, they kind of rewired their genetic code. So we often think of the genetic code as being like universal, which is why we can get bacteria to express human proteins and things, because they all recognize the same codon. So if you stick this in bacteria, a UGU would spell a cysteine, and you stick it in a human cell, a UGU would spell a cysteine. But there is like, it's not quite universal in all organisms because some of them have rewired their codon usage. And so in the case of these methanogens that have this pyrolysine, what they have is they actually have rewired so that their UAG is actually, instead of being stop, it's going to spell pyrolysine. And so they're very, very rarely using pyrolysine when they actually want to say stop. And instead, they're suppressing it and using it instead to put in a pyrolysine. And so when they do have a when they do have a UAG that they actually want to spell stop, normally there's like multiple of them in a row. And then in all the other cases, it's just like normally going to be used for UAG. It's normally going to be used for pyrolysine. So they don't have to worry about all these tricks to kind of trick the ribosome into adding it when it would normally stop, because in these cells, the ribosome wouldn't normally stop there because these have rewired everything already. When we put it in other cells, okay, well, now you might be thinking, okay, well, now it's more of a problem. And it is more of a problem, but it's not a huge problem, because it's this UAG or this amber codon is going to be the most like rare of the stop codons that's used. And it's only like used in about 7% of proteins in E. coli, for example, and barely in anything essential. Also, it's not, if you put it in another organism, it's not going to be totally efficient. And so you're still going to get a lot of release happening at this stop codon. So this is bad if you're trying to like make protein in the lab and it's stopping in the middle of a protein. So you're not getting like the full yield of your products. You're not getting all of the full length protein made. You're getting a bunch of like truncated stuff where it's actually stopping at the release factor, because, well, that's what it thinks it should do, right? You're not in those organisms that are used to using it as in pyrolysine. And so you can get these like truncated products. And so, but on the other hand, you're able to make full length versions of proteins that have the stop codon, or you're able to make like good, like normal length proteins that have a stop UAG as a stop codon to actually get it to stop there, rather than like running through and causing all sorts of problems. So you kind of have this balance where you don't get as much as you want. You get some suppression or some like mismade proteins that you don't want, but it's at a low level, so it's kind of like find your balance type of thing. And so there's also other strategies to try to make things more orthogonal. So more like they're not interfering with what's actually happening in the cell. Things like engineering a 4-letter codon so that the ribosome, there's a tRNA that's actually going to recognize a 4-letter code instead of a 3-letter code. And this is going to expand the possibilities, because basically every codon is normally used, all 64 possible combinations of the 4 letters, all possible 3-letter combinations of the 4 RNA letters, U, C, A, and G, are kind of like taken. And so if you want to get the ribosome to use one of these, you're going to have to suppress what it would normally do or switch out what it would normally do. And so if you're doing it in things like bacteria, there's some people that are like trying to actually like engineer, like actually change all of the different instances of the codon. So like remove all of the places where the UAG would stop and things like this. So there's all sorts of biology experimenting going on, as well as changing the machinery and trying to evolve the pyrolysine machinery, that tRNA synthetase, so the thing that's actually going to add the pyrolysine to the tRNA, as well as that tRNA itself, to evolve these both to be to allow them to recognize other amino acids. And so we'll talk much more about this. This is the basic pathway that we're going to see when we talk about the natural incorporation, the natural making and incorporation of pyrolysine. So basically what you have is you have 2 lysines. So lysine was one of those like normal amino acids, one of the 20 main ones. And these are actually, we're going to see, these are actually going to react and merge together and then like ring up to form this weird thing called pyrolysine. And so basically it's like this pyrrole group and stuff attached to a lysine, so we call it pyrolysine, and it's abbreviated PYL, which is confusing because I always think it should be PYR, but it's PYL, but then I mislabel things, and yeah, so hopefully I fixed all the instances and you don't see any typos, but yeah, so PYL. And basically there are these 5 proteins that are needed in order to make 3 that are needed to make it, and 2 that are needed to stick it in proteins. And so if you just want to stick it into proteins and add like pre-made amino acids, then you just need these 2, these PylT, so the tRNA, and the PylS, this pyrrolysine tRNA synthetase. Sorry, this PylS gene makes this PylRS, so this is the pyrolysyl tRNA synthetase. And this is what's going to load the pyrolysine that you made using these PylB, PylC, and PylD onto the tRNA to make your pyrotRNA synthetase. And this is going to have a stop codon that is going to, it's going to have an anticodon that matches the codon for the for UAG. So it's going to have an AUC that's complementary to that UAG, and therefore allowing you to recognize and that stop codon, bind to that stop codon, and then instead of having a stop, having the release factor come, you're going to have the pyrolysine inserted. And so if we think about where the specificity for this pathway is coming in, it's really coming in from that pyrrole tRNA synthetase, which has to recognize the tRNA, which has to recognize that pyrolysing tRNA, so that tRNA that has that anticodon for the UGA. It has to recognize that tRNA, and it has to recognize the pyrolysine, and it has to incorporate the two together. And it has to do so without messing up what's already in the cell. And so we talk about this as being orthogonal. And so when we talk about orthogonal, that's kind of like something that's happening in parallel and in harmony with the other stuff that's like also happening, but they're not messing with one another. So recently, Carolyn Bertozzi won a share of the Nobel Prize in Chemistry for her work on bioorthogonal chemistry, which is basically allowing you to do chemistry inside of cells and things. So it's kind of like chemistry and biology that play nice with one another. And so with the chemistry, you can do chemistry that's not going to, chemical reactions that aren't going to mess up the biological reactions going on. And those biological reactions that are going on aren't going to interfere with the chemical reactions that you're trying to do. And so often orthogonal chemistry involves the use of like click chemistry, which is what the other two, so Morton Meldal and Barry Sharpless, what they won, their shares for the Nobel Prize was for this click chemistry, where basically you're able to add a group on through bioorthogonal chemistry. You're able to like add a group onto a protein or something, and then react it after the fact or later with some sort of reactive group that's going to recognize basically you tag a protein or something with this reactive group, and then you can add something that reacts to that and get them to react, and like this click reaction. So kind of like a seatbelt where you have two different things reacting and react together, and it's really specific. So it's not going to react with anything else. It's going to react with where you put that tag. And where you put that tag, well, you can put that tag into proteins using non-canonical amino acid incorporation, which you can do with the help of this pyrolysine system. And what makes this pyrolysine system so adaptable is really this pyrolysine tRNA synthetase. So a tRNA synthetase, or an amino acid tRNA synthetase, and its like full name, an AARS, basically it's going to be the thing that adds the tRNA, that adds the amino acid to the corresponding tRNA. And in the case, we call it amino acyl because an acyl group is basically where you have this carbonyl carbon, so a C double bonded to an oxygen, bound to a carbon containing group on one side and something else on the other side. Could be a carbon, could be something else. But in this case, we call this like an amino acyl tRNA and an amino acyl tRNA synthetase because you've got like that acyl group. Yeah, okay. But anyway, this is going to, this amino acid tRNA synthetase, it has to be able to recognize the tRNA and recognize the amino acid. And some of these tRNA synthetases are super duper picky, and they'll recognize if something's wrong, and they can like proofread if they accidentally put in the wrong amino acid or put the amino acid on the wrong tRNA. They're able to like proofread. But the pyrolysine tRNA synthetase is not like that. And so it's able to basically, you're able to trick it more. So you can trick it into adding different amino acids. And this also basically, when the ribosome recognizes this codon, it doesn't make that specific of interactions with this anticodon. So you can actually you can modify this tRNA to actually work with a different anticodon. And so people have done things like make phenylalanine modified versions where it's recognizing putting in different versions of phenylalanine. And so these, in addition to having it so that you can use all sorts of different natural substrates for this tRNA synthetase, because it kind of has that, it's not as able to proofread itself that well. And it has like a big pocket where the pyrolysine binds and stuff. So it's not that specific for pyrolysine. So you can make all sorts of modifications and get those incorporated with just the native PylRS, so that native tRNA amino acid synthase.
But you can also evolve this. So you can evolve that tRNA, that pyrolysing tRNA, as well as evolve that tRNA synthetase. And so this allows you to put in all sorts of different non-canonical amino acids. So here are some lysine derivatives, some phenylalanine derivatives, some alpha hydroxy acids. So all sorts of different things that can be done, various ways to evolve these in the lab. And this is really cool because it way expands what we can think, what we can do, what we have in terms of experimental tools. So basically, when it comes to studying what different amino acids do in particular proteins, it's hard. We can make mutations in the genetic sequence for making a protein. So if we're making that protein like recombinantly, so if we're sticking the instructions for making it into cells, and then getting those cells to make it for us, such as like if on a plasmid that you put into bacteria, we're able to change the sequence and therefore introduce like point mutations, like doing site-directed mutagenesis. We can also mutate the natural sequence inside of cells with things like gene editing. But we're restricted to changing to what's already there, what's already in the toolbox. So we're restricted to changing things to one of these 20 amino acids. And if we want more than just those 20, then we need to be talking about expanding the toolbox with tools like this bioorthogonal chemistry, like this non-canonical amino acid incorporation, where we're going to be able to incorporate all sorts of different things, and we're able to incorporate them in a site-specific way. So if we have ways where we can like add modified versions of amino acids and get them to be used. So this is often used to, as we'll see later, with like radioisotopes. So basically different versions of the atom that are maybe like heavier, and so we can detect them, or maybe they're radioactive, and so we can protect the radioactivity. But you add different versions of them that kind of act the same. So you can have like heavy carbon and heavy nitrogen and things like this. This is used in a lot of the classic experiments in biochemistry, like the Messelson-Stahl experiments and the Hershey and Chase experiments. Basically, you can grow these cells on media, so in food that has the heavy or the light version of these different nutrients, and then they'll incorporate them. But they'll incorporate them into all of their things. So if you add labeled nitrogen, you're going to get all the nitrogens in your proteins and in your DNA labeled. You can do things that are more specific, like just have the labeling on one amino acid, starve the cells, like deplete the media or deplete the food of like the normal version. So like say the normal methionine. And then you could add in something like a heavy methionine or maybe even a selenomethionine, which is really useful in crystallography. You can add these, but then it's going to be incorporated instead of methionine or instead of whatever else you replaced it for in all of the proteins, in all the locations that it shows up. And so this isn't very helpful if you want to study like at a specific spot. And so at a specific spot, then we need to be doing things like this non-canonical incorporation to actually get the modification made exactly where we want it. And since we are doing this non-canonical stuff, we can add non-canonical amino acids. And so we can change and add all sorts of different things that are going to give us all sorts of different functionalities. So all sorts of different things that we can do at those sites. We can also use it to kind of like mimic natural modifications. So those like natural post-translational modifications that happen, it's actually really hard to study them because there's a lot of different, study them in the test tube and things, because these modifications can happen at a lot of different sites, and trying to isolate just like the one version with one modification could be really difficult. And so there are some ways with things like click chemistry that you're able to kind of modify these amino acids, modify the proteins at those specific locations by taking advantage of placing in like a bind here or a like modify here type of signal with these non-canonical amino acids. So all sorts of really, really cool stuff that you can do with these non-canonical amino acids. And so some of these systems that use non-canonical amino acids are based on other things. but a lot of them are based on pyrolysine. So this PylRS, so adding in this PylRS and this tRNA pile. So this tRNA that recognizes the pyrolysine. Remember, this guy's kind of not that picky and kind of sloppy, and so we can get it to add things other than pyrolysine. And so we add the pyrolysine, instead of adding pyrolysine, we add whatever we want them to put instead of pyrolysine, and then we add the pyrolysine tRNA and the pyrolysine tRNA synthetase. And we can either be doing this inside of cells, so we give them in the instructions for making these. Remember, we just have those 2 genes that we need in order to make these, and so we could stick those into cells and get the cells to make them themselves. Or what we could do is we can be working in some sort of in vitro system. So something like a pure system or something like that, where we're doing cell-free protein expression. And then we can just add those proteins like pre-made into this like lysate mix, or I guess not lysate mix in that case. But you can do it in a lysate, or you can do it in this like purified system. And so you just add like the purified components of these. And then you're able to get this unnatural amino acid incorporated into proteins. And so there's been a lot of work trying to evolve above a lot of different versions and different modifications and things that you can add. And so now I want to talk more about how this pyrolysing system was actually discovered, because it's really cool, as well as how it actually works, some of the mechanisms that these machinery is using, both the stuff that we use, the pyrrole tRNA and the pyrrole tRNA synthetase, as well as those other 3 genes that they use in order to make the pyrolysine. Pyrolysine was discovered by Joseph A. Krzyzewski in collaboration with Michael Chan. So they're both at Ohio State University. And so Krzyzewski and his lab were looking into how these archaea called methanogens do their methane making. So methanogens, basically they're making methane. As I mentioned before, this can be really, really useful for like breaking down waste and making methane as for gas and things like this. And so these are often studied. And when they're being studied, they discovered that they had this methylamine methyltransferase, which is one of the components that's needed in part of this pathway for producing methane from these. And what it does is it removes methyl groups from these things called methylamines. So when we think amine, nitrogen, hydrogen, and then a methyl, CH3. And what this enzyme does is it passes it off to other proteins from a methylamine. It takes the methyl group off and passes it to something else to do something with. And when they looked at the instructions, when they found that gene, they looked at the instructions and they're like, wait, this is weird. We know it's making a full-length protein, but right smack dab in the middle of that protein, there's a UAG, which normally would be like a stop codon. And what's more, they found this gene was present, like the stop codon in the middle of this gene was like present in like a bunch of other different, in other like homologs and things, like it was conserved. And so they're like, okay, this isn't just like a typo in this one cell. There's actually these organisms seem to actually be incorporating an amino acid into this when there's a stop codon. But at this point, they didn't know what that amino acid was. They just knew that these buggers were making like a full-length product. So then it was like, okay, well, what is it that they actually, what are they incorporating? And it turns out this is actually really, really hard for them to find out. And what they did was really cool. So they turned to some structural biology help. And so structural biologists try to figure out like the shape of proteins and the organization of atoms inside of molecules by doing things like X-ray crystallography and cryo-electron microscopy, which are basically ways that you can try to get a look at the structure of a protein, at where all the different atoms are. And if they were able to solve a structure of this protein that contains this mystery amino acid, then they could kind of see what the shape looked like, and then kind of try to figure out what was actually there. And so they collaborated with Michael Chan, and he solved the crystal structure of one of the 3 weird methyltransferases. So there's multiple methyltransferases, and this one's like the MMA methyltransferase, MTMB. And then they figured out the structure of it based on that, as well as some like mass spectrometry and things. They were able to figure out what the structure of pyrolysine was. They were able to figure out what that weird thing that was inserted was, and name it pyrolysine because of that whole like pyrrole ring on the lysine. But then they still had to figure out, well, where did it come from? And so they were able to use labeled lysine, like we were talking about before, and I'll get more into it in a second, where you basically have like a heavy version of lysine. They fed the cells this heavy lysine, and then they found that that it was just the lysine, like all of the molecule, the pyrolysine had the heavy versions of the nitrogen and the carbon that were in that lysine. And so this was telling them that it was just the lysine that was being used to make it. So this is being made from lysine. And so now I want to walk you through some of the key papers where they actually made these discoveries. And so I'll also link to them. I also want to point out there's some really good reviews on the natural amino acid incorporation that I'll also link to. But it all starts back in 2002, at least the published version of it starts in 2002 with backed like companion papers from Krzyzewski and Chan, the structure paper, as well as a companion paper that actually goes into figuring like the genetics and showing that it's actually which genes are responsible for this. And so basically in this structure, this is the structure of that methanogen protein that I was talking about, this methylamine methyltransferase, MTMB, from Methanosarcine bakerii. I don't know. I'm really bad at pronouncing things. I'm sorry. Okay, but anyway, they saw the structure of this protein, and here is modeled in. You can see this pyrolysine. And well, what you actually see in the structure instead, this is just like a model that they put in there, but they were able to model it in because they got this really, really beautiful electron density. So this is from an X-ray crystallography structure, and basically what you get is the location of the electrons rather than the location of like the actual atom. I mean, like, well, the electrons are part of the atoms, but basically you have the protons in the center, and then you have the electrons around them. And so what you're seeing is all like the electrons, which is what's going to be giving you the signal in X-ray crystallography. But then you place the model into it. And the better your signal is, the higher the resolution, the better you're able to make out what it actually is. And so they were able to kind of piece together what was the structure of this, and then use like mass spectrometry to verify once they knew like the kind of rough idea of what they should be looking for. And they were able to then also show with this companion paper that this pyrolysine, the genes responsible for this pyrolysine, and that you have this kind of cassette where you have those 5 genes. And they were able to show that one of these was actually, when they probed, so this is like a northern blot, so basically they're probing, they're taking all the RNA. In this case, it's just the tRNA. In this case, it's like all the RNA. And they're probing to see where they have a sequence that's labeled and that's like radioactively labeled, and that's complementary to the sequence that they want to look for. In this case, the pyrolysing tRNA. And they were able to show that it gets processed into mature tRNA. And it's also processed, it's also made as part of a long precursor. So all these are kind of like made in one like cassette. They're all made together and then processed. So they were able to show that. I mean, you can see that it's right next to the genes that have this, these methyltransferase genes that actually contain this UAG. So it's TAG here because this is showing you the DNA and the gene. And so they were able to take a sequence that complemented the sequence of the pyrolysing tRNA and probe for it in all the tRNA as well as in all the RNA, show that it was actually getting made into tRNA, that it was made as part of this larger cassette. Now they wanted to actually show what these genes were doing, and that they were actually having the activity that they predicted based on just like the sequences and similarity to other things. So what they did was they took the gene for making the PylRS, this PylRS gene, and they stuck it into E. coli. And they wanted to get the E. coli to make a lot of the protein for them so that they could purify out that protein and test it in vitro way. So like in a test tube where there's not all the other stuff interfering. So they took this protein, they added a his tag onto the end. So basically a little tagged trail of histidines, the amino acid histidine, to help it purify. They purified it out of the E. coli, and then they mixed it with tRNA. And so they mixed it with either tRNA from the archaea, so it would contain that pyltRNA, but also all the other tRNAs. And it was only like a crude mixture. So, I mean, there could be other stuff in it that could be contaminating. So it wasn't like, it wouldn't be a slam dunk if they found the activity here. But here, what they did was they took that pyltRNA gene and they actually in vitro transcribed it. So they made that tRNA like in a test tube and purified it so that you don't have like any other contaminating things. And so you could only say, if you see activity when you mix this enzyme with this tRNA, you can say, yes, this enzyme is able to add the lysine onto the tRNA. And we're talking about lysine because at this point they didn't, they were still trying to figure out what pyrolysine was. And even once they figured out what it was, well, they didn't have a way to produce it in vitro and produce it like in the lab and stuff like this. They didn't have a synthetic route for making it. They didn't really have a way to purify it yet. And so basically they went with lysine, which they knew was somehow related. And when they add the lysine, they're able to find that this enzyme is actually able to incorporate that lysine into that pyltRNA. And so this is helping show how you can actually have these stop codons be read through in these organisms. So they were able to show like what gene was responsible for that. This was actually an active amino acid transferase, and it was active on this pyltRNA. And so this was really important to help show what was going on inside of these organisms that naturally use it. But, well, naturally, the next question is, can you get organisms to unnaturally use it? So could you stick that those genetic information into other cells and get them to do something similar, get them to suppress their amber codons, stop codons? So that was the next question. In 2007 was they followed up, and so Krzyzewski and his lab, they were able to actually take that cassette, and now instead of just like modifying it where it is, they were able to stick it into other cells and get those cells that wouldn't normally make it. So get like E. coli cells to actually put the cassette into E. coli cells. And now the E. coli cells would be incorporating, making and incorporating pyrolysine. And so this really showed that you can transfer this into other cells and get other cells to kind of recode themselves, at least in this, in specific instances. And so this really helped pave the way for using this amber suppression in other types of cells. And then though they still had to show what it was that was actually where this pyrolysine was coming from. And here's a nice review article of this paper from 2011. And here what they show, basically they were able to, they were able to label the lysine. So they were able to use isotopic labeling. And so when we talk about an isotope, this is a version of an element of an atom like that has more or less neutrons than like normal. So atoms are made up of protons, neutrons, and electrons, and the protons are positively charged, the neutrons are neutral, and the electrons are negatively charged. And basically, the number of protons defines the element. And so say in nitrogen, it's always going to have 7 protons, but it can have different numbers of electrons and different numbers of neutrons. We often focus on electrons because those are what atoms like interact with, and they like share pairs of electrons to form bonds and things like this. And if you have an imbalance of protons and electrons, you get a charged molecule or an ion. You can also, though, have a different number of neutrons, and this isn't going to change your charge because neutrons are neutral, but it will change how heavy you are. And so if you have like more neutrons, you're going to be heavier, and if you have fewer neutrons, you're going to be lighter. And this difference in weight can be detected by methods like mass spectrometry, which is kind of like you can like chop molecules up and then see how heavy they are. Well, it's really like their charge to weight ratio, but yeah. But anyway, you can also, so some versions of these are going to be radioactive if you have like too many or too few neutrons. Like they can be unstable, and then they can undergo decay and let off these like energetic signals that can then be detected as radioactivity. But you can also have non-radioactive versions where you can detect them with things like the mass spectrometry, but you're not going to, they're not going to give back radioactivity. But what's really nice about nuclear isotopes is that they don't kind of mess up with what's already going on in there. So the the molecules can't really recognize that there's a neutron, an extra or neutron or anything like that. So it acts exactly the same. So they could actually take this lysine and they could label it. They could add, use like heavy carbon and heavy nitrogen. And when they did this, they were able to then, the lysine that all this lysine that the organism used was going to be heavy. And then what they did was they isolated the pyrolysine that's made by these organisms and then looked to see how many heavy atoms they had in there. And they were able to show that all of the parts were heavy. Basically, all of it was made from the lysine. If it was made from something else, then part of it would be heavy and part of it would be light, but they found that all of it was heavy, which was telling them that it was all made from lysine. And this was important because there had been, this leads to this pathway that we'll look at in a second. But this was important because there had been some thought that maybe it came from something else. So they knew that there was like at least one lysine involved, but there was also like a report that ornithine could be used. And so ornithine, this is ornithine. And basically you can see it looks kind of like lysine, but it's kind of got different stereochemistry. So things are like switching in different directions because this is D and this is L, and this is one longer. But anyway, kind of similar looking. And it was found that this could kind of get incorporated into pyrolysine or something that looked like pyrolysine. In their study, they were actually able to show that you could use ornithine, but if you use ornithine, what's going to happen is you're going to get a different end product. You're going to get this methyl pyrolysine. So you're going to get this pyrolysine that's missing this methyl group, this CH3 group. So it's a different product. And so the pyrolysine itself is actually just being made from lysine. And why does this matter? Well, it doesn't just matter because, well, it's cool to know exactly where it's coming from, but also it helps show that there's some promiscuity in this tRNA, aminoacyltRNA synthetase, this PylRS, because it's going to be using something that's not exactly like pyrolysine. Like this methyl pyrolysine was actually getting incorporated into the proteins as if it were pyrolysine, but it wasn't pyrolysine. And so this was like, oh, hey, this thing's got some flexibility, and this flexibility is going to then allow us to do things like use non-canonical amino acids. And so they were able to piece together this pathway and show that potentially we can use this in other cells, because remember, they put the stuff in E. coli and got E. coli to use it. And we can modify it so that we can use modified versions of it, because they was able to say, take this make this methyl pyrolysine instead of the pyrolysine. It was still able to use it. So maybe there's some flexibility there. And so these studies really paved the way for everything that's come afterwards. So now let's talk in a little more detail about how this process actually works in order to kind of get this tRNA, pyrolysing tRNA. So going back to our comparison to selenocysteine, what happens with selenocysteine is selenocysteine has its own tRNA, just like pyrolysine had its own tRNA. But with selenocysteine, that tRNA, instead of getting loaded with selenocysteine, it actually first gets loaded with serine, using the tRNA, aminoacyltRNA synthetase that serine also uses. And then it gets modified before it's added into the protein. In the case of pyrolysine, it's made before it gets loaded onto the tRNA. So it needs its own tRNA as well as its own tRNA synthetase, but it doesn't have to do any modifications after that tRNA is actually made. And then, so how do we actually make this? So we have to make that pyrolysine first, and then we have to incorporate it load it on. And so we've been talking a lot about this loading, how we have this pyrolysine tRNA synthetase, and this pyrolysine tRNA. And then we have to make it first, though. And so the making it is done by this PylB, PylC, and PylD. So how it works, and remember this is being worked out in those papers that we were just talking about, was that you're going to use a variety of different enzymes. So remember, enzymes are like reaction helpers or speed uppers. So they can help make reactions happen in ways by like holding things together in the right orientation, promoting the reactions. There are lots of different types of enzymes, and in the case, we can classify them in different ways. In this case, we're going to start with a mutase. This mutase PylB. So a mutase is a type of isomerase. So basically, an isomerase is going to help molecules rearrange their atoms. And then in the case of a mutase, it's not just changing like the orientation in space, it's actually changing connections, like linkages within a molecule.
So in this mutase, in addition to moving the group, you're actually also swapping swapping some of the stereochemistry at a site. So you're switching like forward, backwards type of thing.
And so in this case, what it's doing is it's actually taking this, it's moving a methyl group onto here. So you can see that you're not changing the number of carbons, you're just changing the linkages. So you're making it so that instead of having like 5 carbons in a row, you have 4 carbons in a row and then like one kind of branching off. And so how this is going to happen is it's going to use this mutase. This mutase reaction is a radical reaction, so it involves a lone electron. And normally we kind of like want to avoid free radicals because these things are really reactive. But if controlled in the site of an enzyme, they can be really, really useful. And this is going to be done with the help of a SAM cofactor. So a cofactor is kind of like a helper molecule that enzymes can use in order to give them different properties. And so just like we were talking about how non-canonical amino acids can allow us to do different things, well, having these enzymes bound to other molecules can allow them to do things because they have those molecules. And in the case of SAM, well, this is S-adenosyl methionine. We talked about it a lot when we were in the methionine post on methylation, because it's often used as a methyl donor, but it can also be used in these radical reactions. And so here you're using the SAM cofactor in order to help you do this. So you're making this awkward rearrangement. And so here you can see that this methyl group, this is going to be where we get that methyl group sticking off of pyrrolysine. And that's the methyl group that's going to be missing in the desmethyl pyrrolysine, which you can make from ornithine. And so basically you have now you've made a 3-methyl ornithine, and then you are going to combine it with a second lysine molecule. So we're going to take 2 lysine molecules to make this pyrolysine. And to react them, this is going to use PylC, and this is going to require ATP. So we need to spend a little energy money in order to make it. And so how it's going to happen is it's going to rely on lysine's ability to serve as a nucleophile. And so basically lysine is able to form bonds with different things because it's able to act as a nucleophile. So a nucleophile, much more on this in other posts, but basically it has more electrons than it wants. And so it wants to share those electrons with something, and it wants to share them with something that wants them, which we call an electrophile, and much, much more on other posts. But just know that a nucleophile can attack an electrophile to form a new bond, or it can attack and steal a proton and then just become protonated. So normally when we talk about lysine, it's in this charged form because it's stolen a proton. But if it hasn't stolen a proton, well, now it's got a lone pair of electrons that can act as a nucleophile and can attack things like a carbonyl carbon. And so basically what we get is we have these lysines are going to be linked together. Once you have these lysines linked together, you have one more enzymatic step. So we've used the two of those enzymes, but we have the third, the PylD. What it's going to do is it's going to oxidatively deaminate this product. So basically oxidation is the loss of electrons, reduction is the gain of electrons. Oxidation is often, but not always, involved with the gain of a bond to oxygen, and much more on this in other posts. I'm not going to go into it other than to say that this is an oxidation reaction and you're losing an amino group. So this is deaminating it, and oxidatively so, because you're gaining this bond to oxygen, and you're losing electrons. So that was all the enzyme mediated stuff. But now this is a really awkward thing, and so it's just going to kind of like naturally cyclize. So you're going to get it so that you have this cyclization, and so this nitrogen can kind of like attack here because this nitrogen is going to be nucleophilic as well. And this is still an electrophile, so you're going to get this kind of like ringification, and with the release of water. And now you've made that pyrolysine. And now that you have this pyrolysine, well, it can get incorporated into loaded onto this tRNA pyrolysine by the PylRS. And then voila, you've got that tRNA. And now when the ribosome's traveling along and it sees a UAG, well, now it's going to add the tRNA pyrolysine because this tRNA is going to come in, it's going to bind, and it's going to get, the pyrolysine is going to get transferred just like any other amino acid would. And so then remember that this is going to be useful for those methanogens and stuff because the pyrolysine is useful for them. But it's also useful for us in the lab because we can take advantage of the system to get to use the tRNA, pyrolysine tRNA and this pyrolysine tRNA synthetase in order to actually get them to incorporate things other than pyrolysine. And so this is one strategy for non-canonical amino acid incorporation. And it's another kind of just like call. I hope that you can see from this the importance too of like basic research because these research into figuring out what these little archaea were doing and how they do it. This more like basic research, like what are they doing, how and why, not how can I use this for non-canonical amino acid incorporation. It leads to all of these cool effects. So support basic research and things like that. Okay, so I'm going to end there. Sorry, this was kind of long and rambly, and hopefully it made sense. Hopefully it was helpful, and I'll post the links in case you want to learn more.